Hello, my name is Andrew Templin, and I'm an assistant professor at IU School of Medicine and the IU Center for Diabetes and Metabolic Diseases. I'd like to thank the Sugar Science for allowing me to speak with you today about some of the work happening in my lab and also for highlighting the excellent research that's happening in our Diabetes Research Center. So today I'm going to tell you a little bit about novel roles for RIP kinases in beta cell cytotoxicity. As you likely know, type 1 diabetes disease progression occurs over a relatively long period of time, wherein individuals at genetic risk develop an immune activation against islet beta cells, which results in immune responses against these that can be characterized by development of islet autoantibodies. And we now consider individuals with normal blood sugar, but two or more islet autoantibodies to have stage 1 type 1 diabetes. And this will progress into stage 3 type 1 diabetes, which is when individuals are usually diagnosed with the disease. And we're very much interested in the early stages of this disease progression because we think that by intervening in some of the molecular and cellular processes that are happening, we can change the outcome of this disease. My lab specifically is interested in beta cell death and mechanisms thereof. And this is a model that sort of highlights some of our understanding of what happens between the beta cell and immune cell compartments in T1D pathogenesis. So we believe that beta cell stress elicits chemokine release and recruitment of immune cells to the islet, leading to insulitis. This insulitis in itself can then lead to additional islet damage, which can produce beta cell death and also ongoing beta cell stress. This beta cell death has been attributed mostly to apoptosis. And there are two canonical forms of cell death that we sort of understand. The first is necrosis. It is a passive and unprogrammed form of lytic cell death. This could occur, for example, by cellular trauma or freeze thaw of cells. There's also apoptosis, which is considered an active and programmed form of cell death, which is non-inflammatory. And really, traditionally, it's been thought that all signaling that would elicit cell death would occur via apoptosis. So I'm going to tell you a little bit more about these programmed forms of cell death. Apoptosis is regulated in response to intrinsic and extrinsic signals. It's required for development and tumor suppression, characterized by caspase 3-7 activation and membrane blebbing or apoptotic body formation. And these membrane blebs or apoptotic bodies are rapidly phagocytosed in situ, and that makes this whole process non-inflammatory in nature. And so when we treat cells with a classical apoptotic stimuli like TNF-alpha and cyclohexamide, you can visualize some of these morphological features of apoptosis, including this membrane blebbing and apoptotic body formation that would be phagocytosed by immune cells. In contrast to apoptosis, there are several other forms of regulated cell death, which are lytic in nature and have been characterized more recently. One of those is necroptosis. And like the name suggests, it is programmed but lytic in nature. It's inflammatory and immunogenic. It is generally thought to occur when caspase activity is inhibited or suppressed, and it is known to be triggered downstream of certain signaling nodes like TNF-alpha, interferon, and TLR signaling. And it is critically dependent on these related kinases, receptor interacting protein kinase 1 and receptor interacting protein kinase 3. And we sort of became interested in this biology in collaboration with Andrew Oberst at the University of Washington. And when Andrew was doing some work on this, he was able to image necroptotic cells where he treated with TNF-alpha and a pan-caspase inhibitor called ZVAD. And the morphology of this cell death is quite different from what we see with apoptosis and is consistent with this programmed lytic cell death. So what's known about RIP kinases, generally speaking? Well, RIPK1 and its downstream substrate RIPK3 act coordinately and have functions related to cell death and also non-cell death functions. Within these categories, we can sort of subdivide these functions into caspase-dependent and caspase-independent functions. With With cell death, for example, the caspase-dependent functions are related to apoptosis, and caspase-independent functions related to necroptosis, and also utilizes this molecule MLKL. And the non-death functions are known to include IL-1 beta processing and NF-kappa B activation, and are thought to be related to tissue repair, inflammation, and accelerated autoimmunity. And when we sort of started poking around in islet beta cells, looking at these pathways, we observed that treatment of either mouse islets or human beta iPSCs with TNF-alpha and interferon gamma significantly increased phosphorylation of RIPK1. This is an activating phosphorylation at serine 166. We also found that in NOD mouse islets at 20 weeks of age, or in beta cells from type 1 diabetic organ donors, that RIPK1 RNA expression is significantly upregulated. And if you look at sort of run-of-the-mill mouse pancreas sections or human pancreas sections and stain with insulin and RIPK1, you see that the RIPK1 is abundantly expressed in insulin-containing cells in situ. So this led us to the hypothesis that RIPK1 mediates cytokine-induced beta cell cytotoxicity, and this could be via apoptosis or necroptosis or other functions, and led to several questions. The first of these was, does RIPK1 kinase function mediate cytokine-induced beta cell death in vitro? And you may say, well, of course it's the kinase function because this is a kinase, and that's true. There is a kinase domain in RIPK1. The catalytic site of this kinase domain is at D138. However, there are other domains in this protein, including the RIM domain and death domain, that are involved in scaffolding functions of RIPK1. And these are important for RIPK1 dimerization and activation, and also interaction with other molecules, including RIPK3. There are some small molecule RIPK1 kinase inhibitors that have been characterized. The most well-known is NEC1, and we've used a different one called SCM679 that I'll talk a little bit about later. We started off by performing CRISPR-Cas9 gene editing of RIPK1 in NOD-derived NIT1 beta cells. And we designed guide RNAs targeting exons 2 and 3 of RIPK1. And this is some validation of those CRISPR gene-edited cells here on the right. And then we performed cell death assays, wherein we used an instrument called an IncuCyte to perform live cell imaging and analysis every hour. And we quantified the cell death using this cell death indicator called cytox green. This is not specific for apoptosis or necroptosis or any other specific form of cell death, but rather is labeling all dead cells. And so we can add this cell death indicator and then quantify cell death over time as a function of the phase area of the cells in the well. And what we see when we do that is that when we treat the control cells in the circles with TNF-alpha here in blue, we see a significant upregulation in cell death over 24 hours, whereas that is significantly blunted in the RIP1 gene-edited cells in this light blue triangle. These are the images, representative images of that at 24 hours, and then the death, the quantification of the cell death under various conditions here on the right. We also performed a similar study using our small molecule RIP1 kinase inhibitor, SCM679, which was published in 2022, and we subsequently had synthesized and evaluated. And similarly to the RIP1 gene editing, we found that inhibition of RIP1 kinase function with this small molecule significantly blunted the TNF and interferon-induced cell death in these cells, as shown here and quantified on the right. We also looked at caspase activation. So we know that there's roles for RIPK1 in both apoptosis and necroptosis, and we wanted to look at caspase activation in these cell populations. And to do that, we again turned to our live cell analysis instrument, and we used this dye called Cas37 green. It is basically a caspase 3 and 7 substrate that, when cleaved by either of these caspases, can translocate to the nucleus and fluoresce and indicate caspase activation. And so again, the control cells treated with TNF-alpha in the dark blue circles, By 6 hours, we see a significant increase in caspase 3-7 activation, which was not present in the RIP1 gene-edited cell line. We used another approach to quantify caspase activity, this time using a luminogenic caspase substrate that is available through Promega. And then we tested our small molecule RIP1 kinase inhibitor again. And 4 hours after treatment with TNF-alpha and interferon gamma, we had a significant upregulation in caspase activity that was blunted with addition of SCM-679, shown here on the right. We also were able to look at this in intact islets, where we developed a system to quantify cell death. This again is using cytox green in intact islets, in single intact islets in round bottom 96 well plates. So again, we can treat our islets with our stimuli of interest and our cytox green, and we can quantify the cell death in these islets over time as a function of the phase or bright field area of the islet itself. And on the right, here are some representative images in RIP1 replete islets on the top, treated with vehicle, TNF-alpha and interferon, or TNF-alpha and interferon and BV6, which is an inhibitor of IEPs. And we did the same in these RIP1 kinase dead RIPK1 D138N islets on the bottom. So BV6, just to give you a little background, is a SMAC Diablo mimetic, and it is also an inhibitor of inhibitors of apoptosis proteins. And these IAPs are thought to repress RIPK1 in the setting of cytokine stimulation. So by treating with BV6, we can essentially de-repress the RIPK1, and we hypothesize we would get more RIPK1 activation and cell death. And in fact, that's exactly what we saw. So when we look at these squares, so these are the wild type islets, and we treat them with TNF-alpha and interferon gamma, we see a small but significant increase in cell death that's quantified here on the right. And when we add BV6, the IEP inhibitor, to that culture, we elevate the amount of cell death that happens over the first 36 hours, again quantified here. And when we do the same treatments in our RIP1 kinase dead islets, which are in the diamonds here, we basically find no increase in cell death. So this showed us that this TNF and interferon-induced cell death is dependent on RIP1 and is regulated downstream of IAPs. We took this a step further to try to show that this cell death was happening specifically in islet beta cells. And what we did here was we dispersed islet cells from either wild type or RIPK1 D138N islets. We then treated with an AAV M-cherry. This is an INS2 M-cherry AAV. So we specifically labeled beta cells red. And then we could add our green cell death indicator here and do co-localization to specifically quantify cell death in beta cells. And here again, we found that in the wild type beta cells, treatment with TNF-alpha elicited a small but significant increase in cell death. And when we added BV6 to that, we had a significant increase in the degree of cell death that occurred. Whereas when we did that in our D138N beta cells, we didn't see any increases in cell death with cytokine treatment. We also have done a little work with the Indo-C beta H1 human beta cell lines, where we treat with TNF-alpha and interferon gamma. This takes a little bit longer in the human cells, but by 72 hours, we have a significant increase in the cell death with the cytokines. And this cell death can be amplified significantly with addition of the IAP inhibitor BV6, whereas the IAP inhibitor BV6 alone really didn't have an effect on cell death. So again, this suggests that these cytokines are eliciting RIP1 dependent cell death that is under the control of IAPs. This is that cell death quantified in the human beta cells, and it also looks like this cell death that's elicited following TNF and interferon stimulation occurs in association with increased RIP1 phosphorylation. So this is all consistent with RIP1 being important in cytokine-induced cell death, but it doesn't really get to the question of the role of non-apoptotic forms of cell death in that process. And so we tried to ask some questions about that using a small molecule pan-caspase inhibitor called ZVAD. And what we did was we treated cells with TNF alpha and this caspase inhibitor ZVAD. This is in the dark green. And we found that the caspase inhibitor could prevent the TNF-induced cell death in these cells over the first 24 hours. But by 48 hours, we had an increase in cell death that was significantly elevated above the control. Interestingly, we could block that caspase-independent cell death by addition of SCM-679 as quantified here. And this is caspase activity, just showing that indeed the caspase inhibitor was effective at preventing induction of the caspase activation. We took this a step farther and used our CRISPR gene-edited cell line. And so again, we have this caspase independent and TNF alpha stimulated cell death that we can visualize between 24 and 48 hours. When we did that in our RIP1 gene-edited cells in triangles, we could prevent that cell death, shown here and quantified in the center. And when we added BV6, we could amplify that caspase-independent cell death. And this again occurred in the absence of an increased caspase activation. So this suggested to us that this caspase-independent form of cell death, again, was a regulated event that occurred downstream of IAP. So we showed that in principle, the cells don't require caspase activation to undergo cell death, but what about just cytokine stimulation itself? Is that only eliciting caspase-dependent apoptosis, or is it eliciting other forms of cell death? And we tried to get at this using a different approach, which utilized cytox red, which is another cell death indicator, this time in red, and our caspase 3,7 green indicator that I explained earlier. And we could co-label these NIT1 cells with both of these dyes over time in response to TNF and interferon. And by doing that, we could see either cytox positive but caspase negative cells, which are red, and we would call non-apoptotic cells. We can also see caspase positive but non-dead cells here in green. And we can also visualize yellow co-localized objects, which are both caspase 3,7 positive and dead. So we'd call those apoptotic cells. And we did these studies with TNF alpha and interferon stimulation in our control and RIP1 gene edited cells. Images are on the left. And we quantified this over 12 hours. And what we found was that the cytokines did indeed induce apoptotic cell death over the first 12 hours, and that was significantly blocked in our RIP1 gene-edited cells. And similarly, we found that there was an increase in non-apoptotic cell death, so dead cells that did not exhibit caspase 3,7 activation. And again, this non-apoptotic cell death was blunted in our RIP1 delta cells. We performed a similar set of experiments in control cells using our small molecule RIP1 kinase inhibitor, SCM, and basically found the same thing. So we were able to abrogate the cytokine-induced cell death with RIP1 kinase inhibition, both apoptotic cell death and non-apoptotic cell death. So we next asked the question, does RIPK1 contribute to autoimmune-mediated beta cell killing? So certainly this is involved in cytokine-induced cell death in vitro, but what about a slightly more complex system utilizing autoimmune cells? So to do this, we collaborated with Erica Kai and Sylvain Yu at the Indiana Biosciences Research Institute. And essentially what we did was isolate diabetic splenocytes from NOD mice. And then we took these splenocytes and co-cultured them with either NIT1 control cells or NIT1 RIP1 delta cells. The first thing that we did was we performed an ELISpot assay to look at interferon gamma production as a measure of immune cell effector function. And we found that when the splenocytes were cultured either with control or RIP1 delta cells, they produced about the same amount of interferon gamma, which is quantified here, whereas our positive control showed a significant increase in this interferon gamma production. So it didn't seem like the immune cells were being activated very differently by control versus RIP1 gene edited cells. And in line with that interpretation, when we did flow cytometry, we found that CD69 positive CD8 T cells were basically equally activated following co-culture with our control or RIP1 gene edited cells. But interestingly, the RIP1 gene edited cells were significantly protected from death following this co-culture with the autoimmune splenocytes compared to the NIT1 control cells. So again, this leads us to an interpretation of that the loss of beta cell RIPK1 is not so much eliciting differential activation of the immune cell, but rather is doing something intrinsically in the beta cell to protect it from these autoimmune cells. We also used an in vivo model of autoimmune killing to interrogate this question, and these studies are ongoing. But essentially what we are doing is we're taking NSG mice and transplanting our control or RIP1 gene-edited cells on opposite flanks of the mice. We then isolate splenocytes from diabetic NOD mice and transplant those diabetic splenocytes into our NSG mice carrying the grafts. By the way, the grafts also express luciferase, so we can visualize and quantify the abundance of these grafts over time following administration of the splenocytes. And when we do that, we see data that looks something like this. So out towards 2 weeks to 19 days following splenocyte injection, we see that the RIP1 gene-edited grafts tend to be protected compared to the control grafts. And that's quantified here in images on the right. And again, these studies are ongoing currently. So what mechanisms may underlie this RIP1 mediated beta cell cytotoxicity? And, we're interested at this stage specifically on looking at RIP1 kinase function versus scaffolding function. So we utilize this approach called kinome profiling. And how this works is we can take cell lysates and pass these lysates over multiplex kinase inhibitor beads, which are essentially beads that are conjugated to nonspecific kinase inhibitors. And when we do this and we pass the lysates over the beads, active kinases will be bound to these kinase inhibitor beads, whereas inactive kinases can flow through the column. We can then elute off the active kinases from the beads and perform mass spec to get a feel for what kinase activity is changing under certain conditions or with gene editing in sort of a broad way. And so we've done this in NIT1 control and RIP1 gene-edited cells, and we found, of course, that RIP1 kinase function itself was downregulated in our RIP1 gene-edited cells. We also found several other hits here, which include MLKL, an effector of necroptosis, PKR, an upregulation of certain kinases, including IKB, mTOR, ASK, which is apoptosis signaling kinase, and JNK kinases. We also performed bulk RNA-seq on these NIT1 control and RIP1 delta gene-edited cells, and we identified several genes that fall into categories sort of TNF-related or interferon gamma-related genes. Some of these include, of course, RIPK1 itself, NF-kappa B, ASK, and JNKs, some cell death-related molecules like caspase 3 and 7, also RIPK3 and MLKL, which are necroptosis-related genes. Also interferon-related genes, including JAK, STATs, STING, CGAS, and some MHC molecules. To follow up on this RNA sequencing, we treated the cells with TNF and interferon and then looked for a subset of these genes, including caspase 3 and STING. And in both of these situations, we found that these molecules are upregulated by cytokine treatment, and that was abrogated with loss of RIP1. We also performed these studies in our RIP1 kinase dead islets, and similarly found that the induction of several of these inflammatory genes in wild-type islets by cytokines was diminished in RIP1 kinase dead islets. So in summary, we showed that RIPK1 is expressed in beta cells and is upregulated in islets from NOD mice and beta cells from T1D organ donors. RIPK1 promotes both caspase-dependent and caspase-independent forms of beta cell death following cytokine stimulation. We showed that loss of RIPK1 protects beta cells from autoimmune-mediated killing in vitro and possibly also in vivo. The cytotoxic effects of RIPK1 are associated with actions on kinase signaling and gene expression, and we propose that RIPK inhibitors could block inflammation and cell death signaling in the early stages of T1D to reduce beta cell loss and improve glucose homeostasis. And we have a lot of ongoing work that is moving in that direction. With that, I'd like to thank all of the people who made this work possible, my lab members, our funding agencies, and of course, I'd like to thank the Sugar Science again for this opportunity to share our work with you. Thank you very much.